We're going to continue talking about fungi today. So the other day we got started looking at the Basidiomycota, and there are lots of different Basidiomycota, but probably the one that people are most familiar with is mushrooms. And this includes the type of mushrooms that we eat. Notice that, like a lot of fungi, when you look at a mushroom, most of the mushroom body is actually underground. The mushroom is actually the reproductive structure of the fungus. It sticks up above the ground, again, so that spores can get blown on the wind to a new location, like we talked about with some of the molds. But if we look at the mushroom, if it ever rains, you might have mushrooms that pop up in your backyard. If you pull them up, the next time it rains, you still have mushrooms. And you might think, how can that happen? I pulled them up, they're dead. You didn't kill them, you just pulled up the reproductive structure above the ground. Most of the mushroom body is below the ground. So you didn't really completely eliminate it. So from that point of view, you haven't really killed that mushroom, that fungus. So when we look at the mushroom, the part that's underground is the part that feeds. These are the hyphae that absorb the nutrients. And if we look at this, this is the primary mycelium. And the primary mycelium can divide and make hyphae that are two different mating types. And they used to be called males and females, but at some point biologists realized they weren't really similar to what we think of as males and females. So now one is called the plus mating type and one is called the minus mating type. And these plus and minus mating type, the hyphae can fuse. So notice they don't label it on this diagram, but we know that when the hyphae fuse, that's plasmogamy. Think of the cytoplasm. So now we have cells that are N plus N, or 1N plus 1N if you prefer. So these are those heterokaryotic cells. Since there are two nuclei, we could also call them dikaryotic cells. This secondary mycelium, this N plus N mycelium, these cells start to divide by mitosis. And as they divide by mitosis, they make a little structure that looks kind of like a thumb. It's called a button. And this button gradually develops into the mushroom. And if we look at the mushroom, it has several organs. This main stalk is called the stipe. It has kind of an umbrella-like rim up on the top. This is the cap. And then underneath the cap, there are some little lines called gills. They kind of look like the ribs on an umbrella that you can see if you look at the bottom of an umbrella. Now, if we look at these gills, the gills start producing basidia. One of them is a basidium. So that's why these are the Basidiomycota. So here's a magnification of a basidium, and we have the two different nuclei in here from the different mating strains. So one is called blue, one is red. And then what happens is fertilization, which, remember, is also called karyogamy. And so fertilization, the blue and the red nucleus unite. So now we have a purple nucleus. They're trying to be color appropriate, I guess. I don't know. But notice that this nucleus is 2N because we had a 1N nucleus, the purple one or the blue one, and another 1N nucleus, the red one, that united. So this basidium is now diploid. This basidium, inside it, we have what we call the zygote, and it divides by meiosis. So we know that in meiosis, we have 2 cell divisions. And after those 2 cell divisions, we have 4 haploid spores. So now we have these 4 basidia spores that stick out, and they're attached to the basidium. So again, most of the time, if we look at this type of fungus, the body is growing underground by mitosis, either mitosis of the primary mycelium or the secondary mycelium. After we have N plus N heterokaryotic cells. So this part of the life cycle where we have the mushroom, the basidium, is really a very small portion of the life cycle. Most of the time, if you would look at the body underground, you're looking at cells that are haploid. Now, notice when you look above ground, you see some haploid cells, you see some heterokaryotic cells, you see some diploid cells. So you see all sorts of cells when you look at the body above ground. That's part of the mushroom that we normally see. So let's look at some pictures of a mushroom. So here's a view of the underside of the mushroom. So here's the stipe, kind of the central stalk. You can see all these gills, these long lines running from the stipe in the middle out to the edge of the cap. And these gills, if we look along the edges of these, this is where we'll see a basidium with spores. And each gill has a lot of basidia on it. So let's think about looking at this through a microscope rather than a macroscopic view. And if we magnify one of those gills, here's what we see. Now notice, like almost all the photos of microscopes that we look at, this is stained. There are a few mushrooms that are greenish color, but this particular type of mushroom, Coprinus, is not really greenish. It's kind of a beige color. So this is a gill. Here's part of another gill. Notice we can't really see the complete gill on either one of these because we would see the cap on one side and we would see the stipe on the other. And if you look at the gill, alongside the gill, you can see these little bulges sticking out. So this little bulge here, this is a basidium. Here's another basidium down here. Here's another one down here. Now, notice on some of the basidia, we can see spores attached. We can only see 2 spores here. Keep in mind, that doesn't mean that there weren't 4 spores because a mushroom is fairly large. So when we make a thin slice through it to put it on the microscope, sometimes we tear some of those spores loose. So you can see some spores that don't really look like they're attached to any basidium. They're just floating around. And that's because of the fact that we tear this up a little bit when we make the slide. Notice that you can also see some spores, these dark ovals, as well as these green ovals are spores. You can also see some spores that are kind of in the middle of the gill. Keep in mind, this is a 3-dimensional object. Everything is, even when it's under the microscope, these things are 3-dimensional. So even though they're very thin, you could have spores that are underneath the gill. And that's some of the spores that we see here. So remember we talked about Puccinia the other day. That's one type of Basidiomycota. So mushrooms are another type. Notice that there are a lot of members of the Basidiomycota that are eaten. And there are also a number of members of the Ascomycetes or Ascomycota that are eaten. And the Ascomycetes or Ascomycota, their common name is the sac fungi, because as we'll see later, the spores are contained in a sac. And this includes some fungi called morels, which are very similar to mushrooms. It includes fungi called truffles, which are actually considered a delicacy. In some parts of Europe, they have pigs that are trained to sniff out truffles. Remember, most of the body of a fungus is underground, and the pigs sniff out the truffles, which people can then dig up. There are also several breeds of dogs in Europe that have been trained to sniff truffles. And you might think, why would anybody ever eat truffles, knowing that they were underground to start with? But they're actually considered a delicacy. And depending on what type of truffle you want, truffles are actually very expensive. If you look at the cost per pound of some of the rare truffles, people who are experts can tell the difference in taste. If you look at the cost per pound of some of the rare truffles, some of them cost as much per pound as metals like gold. And in fact, in Italy, where truffles are very popular, one of the big problems they have with smuggling and with counterfeiting is actually with truffles. People try to smuggle Chinese truffles into Italy, and they try to pass them off as Italian truffles. And apparently, if you really know your truffles, Chinese truffles don't taste as good as Italian truffles. So this is a big illegal business in Italy. Now, this group also includes some yeasts like Saccharomyces. And Saccharomyces we don't eat for food, but Saccharomyces is used to make a lot of food. Saccharomyces is the yeast that's used when you add yeast to certain products that are baked. The yeast carry out metabolism, and remember that one of the products either of anaerobic or aerobic metabolism in yeast is carbon dioxide. So this yeast is what causes bread or cakes to rise. Yeast is also what's used to produce different types of alcoholic beverages. We take some yeast, we give them a carbohydrate, and we put them in an anaerobic environment. And in an anaerobic environment, when yeast break down the glucose in glycolysis and then fermentation, they produce carbon dioxide. That's the bubbles that you find in champagne or the head on beer, but they also produce alcohol. So all alcoholic beverages are made with yeast. And again, it might seem to us like one yeast is the same as another, but different types of breweries have different strains of yeast that they use, because besides the alcohol, which is basically the same chemical, whether it's beer or wine or hard liquor, the yeast produce some byproducts. And these byproducts are partly what give different alcohols their distinctive flavors. This group also includes cup fungi, like Peziza. Peziza is a cup fungus that's found in this area. Now notice here's a blade of grass. So that gives you kind of a rough size value of the size of Peziza. And really, when we look at the structure of Peziza, if you were laying on the ground looking at it, kind of looks like a miniature bird bath. And then this group also includes the fungal partners of some things called lichens. Lichens are a combination of a fungus and a cyanobacteria. I'm not sure why, but they're always classified with fungi. They're never classified with the cyanobacteria. So let's look at some of these organisms. And let's take this cup fungus and let's slice right through the middle of this. So imagine taking a bird bath and slicing through this. Down at the bottom, we have kind of the base of our bird bath, the thing that connects it to the main body of the fungus, which, like our mushrooms that we just discussed, is actually in the soil. The very top, kind of the inner lining of this bath, the kind of the blackish purple area that you see here, this is an area called the hymenium. And it basically has two parts. It has some things called periphyses that we'll talk about in just a moment. And it has asci. Asci is just the plural of ascus. So this hymenium is all this part along here, where you can see a little bit of maybe pinkish in there. Below that, the supporting layer is called the hypothecium. So now let's magnify this hymenium and see what it looks like so we can see the spores. So if we magnify that, what we see is these long cells, and these long cells look kind of like sacs. So this long cell from top to bottom here, this is the ascus. And if we look at the ascus, some of the ascus, again, are broken open, or some of them may have already released their spores. Remember, we said they could get broken open when we make the slide. But if we count for the ones that are undisturbed, there are actually 8 ascospores in there. Now remember, we just talked about the mushroom and we said the basidium divides by meiosis. Well, the 2N nucleus in the ascus divides by meiosis, but after it divides by meiosis, then it divides by mitosis. So meiosis gives us 4 cells, mitosis gives us 8 after that. So we have 8 ascospores. Now let's look at a lichen. And here's a slice through a lichen. And we can find lichens all over the place. If we were on campus, we would do our campus tour and we would see lichens growing on several of the trees on campus. If you see something growing on a tree bark and it looks kind of crusty, it's probably a lichen. If it's very soft and when you touch it, it's kind of like a mat, like a carpet, that's probably a moss. But lichens tend to be a little bit more stiffer. They have a little more rigidity to them. But if we would look at this, what we see is there's actually a layer of hyphae on one side. There's a layer of hyphae on the other, and then hyphae go back and forth in the middle. So this whole part would be the mycelium. And then in the middle, we have cyanobacteria. It says algal cells because cyanobacteria are also called blue-green algae. And in many cases, what we find, especially for the algae that live in these lichens, they can't survive on their own. The only place we find them in nature, at least so far, is as part of these lichens. So notice this is a good example of something we've talked about on several occasions before, symbiosis. In symbiosis, we have 2 organisms living together and they cooperate with each other. So we talked about endosymbiosis, how mitochondria and chloroplasts evolved. This is not endosymbiosis because one cell isn't living inside another. So endosymbiosis, one cell has to live inside a different cell. Here we have the algae living inside a layer of fungi, but it's not really one cell inside another. Notice that algae have sometimes been called lichens, have sometimes been called an algae sandwich. The bread is the fungus and the whatever you put on your sandwich is the algae. So when we look at lichens, they're very important in nature. These are some lichens that you can find, like a lot of lichens. They can grow in very extreme environments. So they can grow on bare rocks. Now this is unusual because notice, those algae are photosynthesizing organisms. How can they grow on bare rocks? Because those fungi layers around the algae are very efficient at absorbing nutrients. So when it rains, the rainwater dissolves some of the minerals in these rocks. The algae can absorb those minerals. And like we've seen with the fungi can absorb those minerals. And like we've seen with several of the fungi that we've mentioned so far, the fungi share those nutrients, those minerals, those raw products with the algae. They can fix nitrogen, which the algae need. So they provide this material to the algae. What do the algae give them back in return? Some of the organic products of photosynthesis. So lichens are very important ecologically because if we have a volcanic eruption and we have lava rocks just kind of strewn across the landscape, what are the first organisms to grow typically? Lichens. And as lichens die, then organic material accumulates. It starts to make soil. And eventually the lichens can be replaced by small plants like grasses and dandelions and things like that. Not only do we find lichens on bare rocks, as a result of volcanic eruptions, but we also find lichens on bare rocks when we have new islands produced by underseas volcanic activity, where the islands gradually rise above the surface. Lichens are often the first organisms to colonize those islands. And in fact, even though they seem pretty insignificant, it's estimated that about 6% of the Earth's land surface is actually covered by lichens. Now, notice that some Ascomycota cause diseases. Lichens a lot of times live on tree trunks, as far as we can tell. They do them absolutely no harm. But there are some Ascomycota that cause something called Dutch elm disease or chestnut blight. And these are diseases that have killed off a lot of the elm trees and a lot of the chestnut trees. Now, one of the things we see is that some chestnut trees, some elm trees survive these diseases. And the ones that survive are ones that are naturally resistant. So then we try to use these trees and we try to grow more trees from them that will be resistant to these diseases. And if we look at people, the disease called ringworm is actually caused by an Ascomycota. Notice that ringworm is not caused by a worm. It's actually a fungal disease. Now, before we leave the fungi, let's talk about one group of fungi that's no longer used to classify fungi. So notice this group is called the Deuteromycota, or the fungi imperfecti. And this is not a phylum anymore. It used to be a phylum. And I included it in here because I think it's good to get a historical perspective and also to get a feel for some of the limitations we have, again, when we study the biology of things that are very different from humans. This was originally defined as fungi that don't reproduce sexually. The reason that this category is no longer used is because it turns out when we study the organisms in this group and we look at them very closely for a long period of time, it turns out that they do reproduce sexually. But they only reproduce sexually once in a blue moon, and the environment has to be very specific for them to reproduce sexually. So as the organisms in this group have been studied more carefully, they get reclassified. So for instance, Penicillium and Aspergillus, you've probably heard of Penicillium, I'm sure, because it makes the antibiotic penicillin. These are two genera that were originally classified in this phylum. Now they're classified as Ascomycota. Why? Because it turns out every once in a while when environmental conditions are right, they make an ascus and they reproduce with an ascus. Now, why didn't we know that before? Because 99.99% of the time, they reproduce asexually. So if we look at Penicillium, they both have similar structures. They both have a hypha leading up to their spores, and this hypha is called a conidiophore. Remember when we talked about sporangiophores earlier, we said that PHORE ending means a hypha that holds spores. And then if you look at the end of the Penicillium conidiophore, there are conidiospores. So these little round circles. When you look at these under a microscope, they actually look kind of like those skeleton hands that sometimes little kids have on Halloween. And one of the things that allows you to tell it's Penicillium, it would be a little bit of a pain, but if you wanted, you could count all the chains of spores. On the other hand, if we look at Aspergillus, it's easy to tell apart from Penicillium. Now, keep in mind, this Aspergillus has been stained, but if you look at Aspergillus, the spores spread out all over the place. They're not organized into a couple of nice, neat lines like penicillium. And so these spores spread out all over the place. And when you look at aspergillus under the microscope, it looks kind of like a cheerleader's pom-pom or a dandelion. There are just spores coming off the end every which direction. But both of these organisms turn out to be Ascomycota. Now, the discovery that the organisms in this group reproduce sexually has really some important implications for medicine. So think about penicillium for a moment. Penicillium is a mold and it makes penicillin. And penicillin was discovered about 100 years ago, actually. And the way it was discovered is how things often happen in science, through serendipity, through kind of a lucky chance. A scientist was trying to grow bacteria on his Petri plates. And his technique wasn't very good. When you make Petri plates and you put bacteria on them, you try to use what's called aseptic technique. You try to make sure that you only put the bacteria on that Petri plate that you want, nothing else, no contamination. Well, this scientist noticed that some of his Petri plates, no bacteria grew on. And he was about to throw them out, but fortunately for the world, he recognized that the Petri plates that didn't have bacteria growing on them had mold growing on them. And it turned out to be penicillium mold. Now, why does penicillium mold make penicillin, which kills other molds and bacteria? Well, because when penicillium mold is growing on a piece of fruit, that's its food. So when it produces penicillin, it's staking out its territory. Now, there's a problem with using penicillin to treat bacteria. If you take some bacteria that cause an illness like streptococcus, which causes strep throat, but other diseases also, depending on what type of streptococcus. If you take a Petri plate and you put some streptococcus on that Petri plate, and then you put a liquid penicillium mixture, most of the bacteria die, but some of the bacteria survive. So in other words, some of these bacteria are resistant to penicillin. And notice this happens, it's something in their genetics. This happens before they're exposed to penicillin. Some of them can survive this. So what this means from a practical standpoint is, if you have an illness and we give you penicillin, that maybe it kills most of the bacteria the first time, but the bacteria that survive and reproduce and make the new population of bacteria in you are the bacteria that were resistant to penicillium. So the second time we use penicillium, it's not very effective. Because remember, bacteria only reproduce asexually. So if a bacteria that's resistant to penicillium reproduces, all its descendants will also be resistant to penicillin. Now, the problem with this is, since penicillium also reproduces asexually most of the time, that means all the different types of penicillin mold make the same penicillin antibiotic. Not good. So scientists were very excited when they discovered penicillium reproducing sexually. I came into class that day and I was really hyped up, and my class was like, "What are you so excited about?" And I said, "Penicillin reproduces sexually." And they just thought I was kind of kinky or something, I don't know. But here's why it's important. If penicillin reproduces sexually, then we can create the correct environment. We can get this mold to reproduce sexually. And when it reproduces sexually, the genetics change. And if the genetics change, that means the antibiotic that it makes is going to be a little bit different. So maybe that antibiotic will still kill bacteria, but maybe they won't be resistant to it. Before now, if we wanted to get alternate versions of penicillin, we basically had to design them in a lab. You know, think back to what we talked about in 105. We had to figure out exactly what the molecule looked like, and then we had to take a functional group off or add a new functional group. And then we had to test it on bacteria and see if it killed bacteria or not after we made it chemically in a test tube. So it was a very slow, tedious process. And a lot of times we didn't get anything that worked. But if we can get penicillin to reproduce sexually, we can just grow tons and tons of penicillin in labs and then sample all the antibiotics that they make and let them do the work for us. And all we have to do is the last stage of the process. All we have to do is test the antibiotic that they make for us. So right now we haven't quite mastered the process of getting penicillin to reproduce sexually as often as we would like. But hopefully at some point in the future, we'll be able to do that. Yeah, we have limited success at this point, but not enough that it's very economically feasible. So it's still mostly something that we're doing in a laboratory situation rather than industrially. So are there any questions about fungi before we leave them for the day? Okay, if there are no questions, I'm going to stop sharing the screen. Should have evolution scores by now. If you haven't checked recently, they should be up. If you want to get your test back, you can get your test back with corrections on it. On Wednesday, we'll try to talk about the exam a little bit, and I'll try to give you some advice about what you might want to try next time. Are there any questions about anything else that we've covered recently? Oh, the other thing that's posted is the prokaryote questions. Those are not due till next Monday along with the paper. So I'm working through the papers right now. So if you sent me in a sample paper, you should get that back sometime today.
So you said the paper is due on Monday?
Yes, next Monday.
Okay.
Are there any other questions? If there are any other questions, I'll stop.